I am very excited to introduce our third speaker in the Black History Month celebration. Dr. Sharonda LeBlanc is a assistant professor of physics at North Carolina State University. She graduated from the nanoscale science PhD program at UNC Charlotte in 2012. Her lab designed single molecule fluorescence-based assays to investigate biomolecules in action and answer fundamental biological questions. Time-resolved fluorescence methods employed in her lab include fluorescence correlation spectroscopy, FCS, and fluorescence resonance energy transfer to monitor fast biomolecular interactions that drive biological pathways. She is primarily focused on questions related to three distinct systems: SARS-CoV-2 viral RNA processing, ribosome assembly, and DNA repair. She enjoys spending spare time with her husband and two daughters. Take it away.
Thank you, Feather, for the warm introduction. Thank you, Vanessa, Nikki, anybody else who is involved in this effort. Definitely thank you to AJ, and I guess CZI as well for my support. So I'd like to go ahead and share my slides. I think it might be on the... Do I need to swap displays or no? I'm good?
That's great.
Okay. All right. So today I'll be talking to you all about some of the research that I have going on in my lab. My lab is relatively new. I've been an assistant professor for 3.5 years. So I started during the pandemic. So we are, as Feather said, interested in creating single molecule based assays that can allow us to monitor in real time biomolecules and how they're functional in certain pathways. So I've got to get used to doing talks in Zoom again. I've shifted back to in-person. So I'd first like to acknowledge all members of my lab, past and present, in addition to my collaborators and my mentors and funding sources. So I started as a biophysicist, actually as a postdoc. And before that, I was doing single molecule spectroscopy, but on nanomaterials. So I was really interested to find out how enzymes actually used ATP in cells. And so they use ATP to change shape, and that allows them to drive large scale motions and long range effects. And from there, so my lab kind of is really interested in this broad question of how enzymes coordinate over space and time to carry out essential cell functions. And from there, in a more applied context, we're interested in how mutations impact that molecular function, and then ultimately how mutations lead to disease. And so we take a single molecule approach because a lot of what we're interested in, or a lot of the fundamental processes that drive biology, happen very quickly and are dynamic. And many of those things you can only see at the single molecule level. So most of the imaging that I'll show you today is single molecule imaging, and I also have some spectroscopy that goes along with the imaging. So our tool of choice here is a time-resolved confocal microscope. It's a commercial system made by PicoQuant. It gives us access to quantitative fluorescence imaging and spectroscopy. Let me get this thing out of the way. Okay, there we go. So it's like a confocal microscope that you might be familiar with, but we have a few other pieces that make it a little bit different. So the main two things are these avalanche photodiode detectors, which detect individual photons, and that allows us to build up an image. And those detectors are connected to event timers, so that gives us fast temporal information. Looking a little bit more closely at this main optical unit on the next slide, we have all of our lasers fiber coupled into this main optical unit. So this houses all of our confocal optics. The laser comes in here. This is just a mirror. I don't know if you guys can see my cursor or not, but this is just a mirror. And our signal, our laser comes in goes through a couple of filters, and this is our main dichroic. So this is the key part that splits the excitation from the fluorescence emission. So going this way, all of our laser wavelengths are reflected off the surface of the dichroic, and then our fluorescence emission goes through the dichroic. The longer wavelengths go through the dichroic. And so we have a pinhole here that is exchangeable. We can change it to different micron scale sizes. And then we have a mirror here. And in this optics tower here, we can swap out between either a dichroic or a mirror, or we have other beam splitting optics that we can use. And so here's the interesting part that allows us to do a lot of different things with our system. So we have these two detectors, and they're single photon counting detectors. They're not cameras. They allow us to build up a map of fluorescence intensity and also fluorescence lifetime. And so these detectors are connected to this event timer, which I'll tell you a little bit more about on the next slide. But the main takeaway here is we don't use a camera for imaging. We use the single photon counting technique, and we build up a map of the fluorescence intensity and lifetime simultaneously. So how does this work? So we have a really fast beam scanner that rasters over the surface of our image, and it works by time correlated single photon counting. So I mentioned before that we use pulse lasers. And so if this is the beginning of our experiment here, we have a series of laser pulses, and our laser is pulsing at a megahertz repetition rate. And there's some probability after excitation that our sample will emit a photon. And so our event timer is just a very accurate stopwatch that gives us two time tags. It gives us a time relative to the laser pulse rate, which is on the nanosecond time scale, and then it gives us an absolute time relative to the start of the experiment. So this time here is continuous, the macro time is continuous, and here this time is fixed by the repetition rate. So this is again, always on the nanoscale, a nanosecond time scale. So we call that the nanotime, and this the macro time. And so if we make histograms of those time tags, then with the nanotimes, we get something that decays exponentially, and that's related to the fundamental photophysics of our fluorescent molecule. And typically we're using fluorescent dyes. We don't really work with fluorescent proteins that much, although you'll see some examples later on with some GFP and some M-cherry, which you might be familiar with. But this excited state decay is characteristic of different types of fluorophores. And again, it's usually on the nanosecond to tens of nanosecond time scale. And so if we bend those macro times, then we get an intensity versus time trace. Again, that's a little more continuous in time, and it gives us dynamic information about our sample. And so we also can separate based on wavelength using a filter in that optics tower that I showed you earlier. So if all of that is too much, then just take away that we know for every photon that's detected at our single photon detectors, we know when it arrived, we know where in this raster scan it arrived, and then we know which detector it arrived at, which will give us spectral information. And so it'll become clear why all of this is needed to tell us about the different aspects of our biological system. So here's that system again, just in a schematic form. I want to call your attention to a couple of things that I haven't mentioned already. We do our experiments in two different ways. Either we put a droplet of a sample on, a sample that contains a labeled biomolecule, either DNA, RNA, or protein, and they're often labeled with two different colors. So a red color, or a red dye and a green dye. And that allows us to do something called FRET, which I'll tell you about a little bit later. But we can either focus on the very top of this cover slip, which will allow us to image. So we can do fluorescence lifetime imaging, or we can focus just above the surface into the droplet, and we can monitor the stuff that's diffusing through the solution. And that gives us, again, information on different time scales. And I'll go into a little bit more detail and show you some examples of what we get. The other thing that I want to call your attention to is this ability to do pulsed interleaved excitation. So we have 3 lasers, and we typically will use the red and the green to do something called FRET. And what we can do is delay the pulses relative to one another on a nanosecond time scale. And that gives us access to the fluorescence lifetime information and the intensity information simultaneously. And so that nanosecond delay between the two pulses allows us to essentially monitor the donor and the acceptor, or the green and the red fluorophore, simultaneously. So that's much faster than any of the biological processes that we're interested in, which usually occur on the millisecond time scale. So those are the kind of main takeaways. These are the things that make our system a bit different, maybe than other systems that you might be familiar with. So that leads to a range of dynamics across multiple time scales. As I said before, if you bend the nanotimes, then you get the excited state decay behavior. In the sort of intermediate microsecond time scale, you get FCS, which is fluorescence correlation spectroscopy. Tells you about the diffusion of your particles in solution. And then for things that are attached to a surface, we can make these intensity versus time traces that allow us to monitor like conformational changes in individual molecules. So together, this TCSPC, time-correlated single photon counting, and the pulsed interleaved excitation enables multiple things to be measured simultaneously. And so the first few things that I'll show you will be things that we can measure in solution, and then I'll get to the things that we can measure on a surface. So here's just a intensity versus time of molecules that are diffusing through a solution. And so all of the tall peaks are individual molecules that diffuse through the volume, and they emit what's called a burst of photons as they diffuse through. And by analyzing these bursts of photons, we can actually recover single molecule information. So although we are sort of averaging signal, we can go back and recover single molecule information that will give us information that we otherwise would not see. So the first thing is FRET, which we do a lot of. FRET is a phenomenon that where if you have a donor molecule and we excite the donor molecule, if it's close enough to the acceptor, then it can transfer energy from its excited state to the acceptor, and now the acceptor can emit light. And so that interaction has a 1/, distance to the 6 dependence, and so it becomes a very, very sensitive nanoscale ruler. I'll go into a little bit more detail about that in a few slides, but what we get is a distribution. We get a distribution of FRET efficiency, and we can ultimately convert that to a distance. So we get a distance between the molecules that's on the nanoscale. Simultaneously, we get, again, on diffusing molecules, we can get FCS, which is fluorescence correlation spectroscopy. That gives us concentration. We get a diffusion coefficient, which can tell us things like the oligomeric state of proteins or DNA, whether or not we're forming complexes. We can also monitor protein-protein or nucleic acid interactions. And so again, we only have a few molecules in the observation volume at any given time. So this can be considered a single molecule technique. The other thing that we get is the fluorescence lifetime. The fluorescence lifetime is another dimension of fluorescence that I think is often underutilized. Typically, people use the intensity, of course, to report the location, but the fluorescence lifetime is also very sensitive to the local environment, so that can be used in biosensing. The FLIM, so this is an imaging group. So you all might be interested in FLIM, which is fluorescence lifetime imaging microscopy. So when we put our samples on a surface, these are actually individual DNA duplexes. So these DNAs, we've labeled them with a donor and an acceptor, and the color of the image is actually reporting on the FRET via the fluorescence lifetime. And so I'll go into a little bit more detail about that in a couple of slides. So looking at those molecules on the surface again, what these molecules are we've encapsulated just individual fluorescent dyes into a liposome. We just do this via extrusion, and then we can put our liposomes on the surface and probe the individual bright spots. So every bright spot here, again, is an individual molecule that's inside of a liposome. In this case, we have just dye, but we can also put proteins in here and monitor proteins as they're doing their thing and diffusing. So that way the proteins are free to move around inside of that volume. By clicking on an individual spot, we can monitor an individual complex over a long period of time, and it will become clear why that would be of interest to us. And so by bending the nanotimes, we get the excited state decay. By bending the macrotimes, we get the intensity versus time, again, for a single molecule. So I mentioned that fluorescence lifetime is a very sensitive measurement. And while this is not my work and this particular image is not single molecule, but it gives you an idea of what fluorescence lifetimes can allow us to do. So these are salivary glands of an American cockroach. Again, this is not my work, but these salivary glands have been labeled with a dye called MQAE. And this dye is quite sensitive to chloride concentration. And you can see here the color scale is actually not just an intensity scale, it's a fluorescence lifetime scale. So very short lifetime of 1 nanosecond and indicates quenching, and a very long lifetime indicates is unquenched. And so the short lifetime is being the excited state is now being quenched on the left side by the presence of the chloride ion. And here in the low concentration of chloride, it is not quenched. So we have the longer lifetime. And so by employing different dyes, you can sense different molecular environments. So I mentioned FRET as a nanoscale ruler. This is something that we used a lot to monitor conformational changes, especially in proteins or enzymes that use ATP binding and hydrolysis in order to change shape. I kind of introduced this before, but we excite the donor with our green laser, and then from the excited state, it can transfer energy. So here's just the equation that goes along with that. If we plot the FRET efficiency as a function of distance, then we get a decay behavior that looks like this, whereas if we're on this very steep part, then we're quite sensitive to the distance between the donor and acceptor. And what we do with that in a biological system is we can put a donor on one part of our sample and the acceptor on the other, and we can monitor interactions between the two. One story that I'll tell you, we'll use the intraprotein labeling strategy where we put a donor and acceptor on different domains of a protein and we can monitor conformational changes. And then another story, if I have time, will be about how we can monitor the cleavage of an RNA, a piece of RNA by an enzyme. Does anyone have questions? I know it's a lot of information. Or should I just keep going? I know this is being recorded.
Yeah.
Okay.
Yeah.
Okay. All right. So some of the biological systems that we are interested in are shown here on the slide, and along with the folks in my lab that are working on these projects or leading these projects. So I have 2 postdocs and a graduate student and a couple of postbacks that I'll show a bit later. The first project that I'll talk about is ribosome assembly. My postdoc Alex is leading that project. Then I'll talk a bit about the mismatch repair project, and then I'll talk about the NSP15. So ribosome assembly, before a ribosome is capable of making proteins, it has to be assembled. And this project and this process starts in the nucleolus, and the small and the large subunit are exported separately into the cytoplasm and they come together. Well, this process is long and it's challenging, and it involves lots of assembly factors and also several ATPases. So several AAA ATPases are involved, including RIC7, RIA1, and DRG1. And so this is a crystal structure from my collaborator of RIC7, which is the earliest acting AAA ATPase in the pathway. And it's thought to use this translocation mechanism to unfold other proteins. And so what we want to do on the single molecule level is be able to monitor this happen in real time, and we're getting there. We're not quite there yet, but we want to actually see this protein in action. But we started by trying to monitor interactions in solution. So there's some evidence that RIC7 unfolds NSA1, which is just an assembly factor in a pathway, in a long pathway of over 200 factors, like I said, that are that have to come in sequentially and be removed, do their job and be removed. And most of these, and all of these AAA ATPases are involved in removing one or more assembly factors. And so we started off by trying to label these two proteins. So we express and we purify these proteins, and then we fluorescently label them, and then we can do FRET with them. So preliminarily, we've labeled this RIC7 with an acceptor molecule, and we've labeled the NSA1 with the donor, and then we can monitor interactions while they're diffusing in solution. And again, this is quite preliminary. Without any ATP, there we see very, very few FRET events, which is the histogram here. With ATP, we see an increase in the interactions between the two molecules. And so that tells us that they're interacting. Doesn't that necessarily tell us how? And we're trying to do some surface attached single molecule experiments that will help us to actually monitor this process happen. We have a little bit more preliminary data that suggests that there are some shape changes with NSA1 in the interaction, which suggests unfolding. So on the imaging side, we're also working with some Saccharomyces cerevisiae that my collaborator's postdoc has made. So she's made some strains of Saccharomyces that have different ribosome assembly factors labeled. In this case, NOP7, which is an assembly factor, is labeled with GFP, and RIA1 is labeled with mCherry. So those are a FRET pair, and again, those are fluorescent proteins. So you might recognize those as being fluorescent proteins. But if we excite them in this pi mode, then we can monitor them essentially simultaneously and ultimately get FRET to see if these two molecules are interacting in a living cell. And so that's what we've done here. RIA1 is thought to remove NOP7 from the pre-6DS particle. So we collect this image in pi mode, and then we do some calculations. This is the equation. I won't go into it, but this is the image that we get when we do a calculation, and we do some corrections to account for any leakage or any direct excitation of the two different fluorophores. But I don't know how well you can see it. I'll zoom in a little bit on the next slide, but you can see some little pixels that are colorful there, and that's where the FRET is happening. So you see lots of color here and not as much color here. So not everything, not all of the complexes are fretting with one another is the takeaway there. Zooming in again on that FRET image, on the bottom here is a histogram that shows the distances that we get with the calculation. So we can see that we get, and the units here, I apologize, are not in actual distance, but these are nanoscale distances that we're calculating. But if we zoom in on individual cells, then we can see, again, these are also living cells. But these are snapshots in time of what might be happening. But one thing that you can see is a different color in different parts of the cell. And what that tells us is that these two molecules are indeed close enough to FRET, so within 10 or fewer nanometers of each other. So they're close enough to FRET, and a different spatial pattern for different conformations of the complexes. So very, very close to the nucleolus, we see this blue color, which is indicative of a low FRET. So the two domains that are labeled are farther apart, and then higher FRET out in the cytoplasm. So we're still trying to understand. This data is pretty new, so we're still trying to understand structurally what that means for the interaction between RIA1 and NOP7. But we think that these spatial patterns make some sense that they would be different in the nucleolus versus in the cytoplasm. So the other thing Sorry about that. So we can zoom in a little bit more, and this is an image that has a few more pixels in it, so you might be able to see a bit more here. So we can again see that different spatial pattern that shows different FRET, so different conformations of the complex near the nucleolus versus in the cytoplasm. So we can also do, so that was intensity-based FRET. So we can also do fluorescence lifetime-based FRET. And we started with a very simple, a simple system, which is DNA. So we have this DNA that we've labeled with a donor and acceptor, and we can attach it to a surface. And so when we do that, we get images that look like this. So every bright spot is a single molecule, and we can look at both the intensity and the color of the individual spots to tell us about the FRET. So the FRET, again, can report on distance. It can report on conformation of the individual complexes. And again, we can probe individual complexes by clicking on a spot and getting an intensity versus time trace, and that's shown here on the right. We put those same molecules in solution, and we can probe the FRET in solution as well. So these are the same molecules or the same substrates, and we put them in solution, and we get a histogram. We get a histogram that results from thousands of molecules diffusing in solution over the course of a few minute acquisition. And the FRET that we get is consistent with the distance between the donor and the acceptor molecule in solution. So just two different ways to measure the same thing. Hopefully you can see that the solution-based method gives us good information, but it's higher throughput. So we in the phlegm, we have to click on individual spots. Here we're getting a lot of information, but there are limitations for the solution-based measurement. So we can get FRET, and then we can ultimately convert that to a distance. So this is an example of how we also use spectroscopy. So these are, again, those dye, the encapsulated dyes, and we're monitoring them as they are diffusing through solution. In this case, again, it's just a simple dye molecule, and every tall peak is an individual molecule as it diffuses through the confocal volume. We perform what's called an autocorrelation on the signal, and the decay of that signal tells us about the size of the molecule. Won't get into a lot of detail about that, but if you have questions, I'd be happy to answer them later. And so we fit this autocorrelated data to a model, and we get some parameters out. So one of those parameters is called the diffusion time. And that is actually characteristic of the size of the molecule and sometimes the shape of the molecule if we're talking about DNA or if we're talking about proteins or DNA. So we did this with a few different sizes of liposomes, and you can see that we get different decay behavior that's consistent with the size of the liposome. So we can make these liposomes in different sizes, and we ultimately fit our diffusion coefficients that we get. So D is the diffusion coefficient that we get, and we can fit that to a theoretical model, which is the Stokes-Einstein equation. And that ultimately tells us that we're getting, reliable information about the size of the molecule in solution. And so if we have a protein that is sort of globular in shape, then we can ultimately get a diffusion constant that can tell us about potentially the interactions that it's having, maybe the oligomeric state of the protein. So we can take that measurement, so the FCS measurement that I showed you with the decay, we can take that measurement into living cells as well. In this case, this image, these are S. pombe cells, and the tubulin is now labeled with GFP. And this GIF here was created from multiple scans, multiple raster scans together. And then we can click on an individual spot. So after we collect our image, we can click on a spot inside of the cell and just kind of sit there and collect photons. So when we collect those photons, we can do that same sort of autocorrelation, fit it with the model, and then get a diffusion coefficient from from that. So this gives us a diffusion coefficient for tubulin during different phases of the cell cycle. And so that's what we're trying to do with in collaboration with my colleague Mary Elting here in my department. So on the method development side, you can also get diffusion from an image. So that is one way. I showed you one way by clicking on an individual spot, monitoring molecules as they diffuse through the confocal volume. But another way is taking that stack of images that you have, the stack of raster images that you have, which contains temporal information. And you can do autocorrelations on the image. And it's something called raster image correlation spectroscopy, or RICS, and you can get a diffusion coefficient from that as well. So this is a postback in my lab. He is able to do the RICS analysis, and he also created something called M6. And we're working on a publication on that now, but it takes advantage of our ability to, instead of doing a raster scan, we do a bidirectional scan, which gives us information in both directions, so forward and backward. So on to some of the biological systems that we're interested in. One system that we're interested in is mismatch repair. Mismatch repair is central for all organisms. Anything other than a Watson-Crick base pairing is considered a mismatch. In this case, I'm just showing you mutase, which is the protein that initially recognizes a mismatch, and then it signals for repair. It calls mut-L to the site, and they interact in an ATP-dependent fashion to coordinate the rest of repair. So we're interested in what are the steps that lead to the initiation of repair, and how do they coordinate the rest of the process. So the rest of the process involves, of course, excising out the damaged part of the DNA and then repairing it further down the pathway. So we're interested in the initiation, and we're trying to understand the molecular mechanism of that, mainly because mutations in both of these proteins are linked to several cancers, including colorectal cancers. So we like to see, how are these mutations ultimately, how do they change the dynamics of these proteins? So previously with turf microscopy, so you can do FRET with turf microscopy if you're familiar with that. It's a surface attached FRET experiment. We did these two experiments where we put donor and acceptor on DNA, and we can monitor the DNA bending state, and we can monitor the protein its proximity to the mismatch here. So we did both of these experiments with turf microscopy. This is turf. If you're unfamiliar, it's different from the confocal in that it's a wide field technique. So we get an image of lots of molecules at one time, but then the post-processing is quite slow because we have to go back, identify donor and acceptor from the images, and then we have to create these intensity versus time traces, and then we ultimately calculate FRET from that. And so it's quite powerful if you want to see sequential events. And so that's something that we got out of those two experiments that I'll show you. So we do the FRET calculation as shown there. But what we did from literally analyzing thousands of single molecule binding events was, well, we saw signatures in both of these experiments, and we wanted to see if we can connect them by their kinetics. And so the picture that emerged was that in about 30% of events, we could see the following signatures. So with the DNA bending experiment, we could see the DNA proceed through a series of bent states, and we could also see the protein proceed through a series of conformational changes, and also see the bent state from this particular label site. So what that tells us is that these events are very short, right? So this whole event lasts for just about 5 seconds or so. So these are events that you would not otherwise see with ensemble averaging. So this state that we saw, this bent state 3, is the protein, the DNA binding domain opening, and the DNA unbinding, which we think is a very key regulatory step in the pathway. It's also where we think mutL might interact. So our next step was to figure out what mutL was doing and how mutS and mutL are interacting. This is quite complex, but this is published. With kinetics, we were able to connect 4 different measurements, previous measurements, and some measurements that I did, but we were able to see signatures from multiple binding events of that bent state or that unbent state where the DNA binding domain opens and the DNA unbends. So again, we think that's a really, really important step. That's probably a final verification of the mismatch. And so mutL, we think that mutL also interacts here. And we're quite interested in mutL and its function. So these are some AFM images that were previously published. This is human mutL, and you can see that the protein adopts these 4 different conformations. So the question is how are these conformational changes functional in mismatch repair? They have to be regulated in some way. ATP is required for mismatch repair, so we think that it's ATP binding and hydrolysis that actually drives this forward. So what we did in my lab was to label mutL in different places, and we monitored the conformations that we got from solution. So the question is, can we recover conformational information from solution using our pi-FRET technique? And without getting into a lot of detail here, we get distributions that look like this, and we can fit our FRET distribution to multiple Gaussians in order to monitor populations. So you notice there are 4 Gaussians here that we fit to, which was actually the minimum number that we could use. And that coincides with the 4 conformations of mutL that were previously seen with AFM. So we assign those peaks to, so the condensed state is high FRET, and then the extended state is low FRET, and we can monitor, again, from solution, we can monitor how those populations change when we add nucleotide. So that's what we did. So this is without nucleotide. When we add ATP, the main thing that changes, or the largest change that we see, is this species 3, which we think is the one-arm state. So this one-arm state, as I'll get to later, I think is critical for mutL reeling in DNA is ultimately what we think is happening. So the one-arm state changes quite a bit. The population of the one-arm state changes quite a bit when we add ATP. We can look at changes with ADP. In the presence of ADP, that the species 3 or the one-arm state seems to be depopulated. With a non-hydrolyzable analog of ATP, which is ATP gamma S. There are changes all over the board, but the main thing is that we see a shift to higher FRET, which indicates that this is the protein is in a very closed or compact state. We do this with different label sites, and we see different distributions. So this indicates to us that we're able to monitor conformational states from a solution of diffusing molecules, which is quite powerful if you want to challenge this assay now with different perturbations. The other thing that I haven't mentioned is that mutL is intrinsically disordered. So it has these intrinsically disordered linker arms, and there's really we don't have any crystallographic information about it. So this is actually a nice way that we can get some sort of structural data from a solution-based measurement that doesn't exist otherwise. And so when we add the gamma S, we can see that the linker arms actually bow out quite a bit to the point where we get 0 FRET. So they're very, very far apart. The donor and acceptor are not fretting with one another, at least not very much in this closed conformation. So how can we perturb this system? We introduce a functional mutation, which is E28A. This is Thermus aquaticus, but when we map it to the human protein, it lines up with E34A, and this is a Lynch syndrome mutation, so linked to colorectal cancers. And we kept the S to C, the N-terminal label sites, in order to report on the distance between the N-terminal domains. So we have our standard distribution here, which with our 3 conformations, and when we add ATP to the mutant, we see that that state completely goes away. So it seems like this mutant that is linked to, that maps to a disease, disease-related mutation. It seems like this protein is now unable to close, it's unable to form a closed structure. And the one-arm state is particularly impacted, and then everything is just really shifted down to low FRET. So it just seems to be unable to close. This is not my work, but my one of my mentors, her lab can do high-speed AFM now. So the previous images that I showed you were static surface-based images, but now they can do high-speed AFM where you can see this protein cycle through conformations in solution. And so this is the human protein. And you can see at some point that the protein, even in isolation without any ATP around, at some point cycles, and it appears to reach out and grab something. So you can see that's the one-arm state that we think is important for reeling in DNA. So there's some evidence that S and L together can compact DNA, and that is likely important for the downstream repair. So in conclusion, we can detect the occupancy of conformational states from solution on these diffusing enzymes, and it's a relatively high throughput method compared to surface attached FRET experiments. And we now have a platform where we can add perturbations on the so in disease-related mutations or potentially inhibitors. When we add a mutation, we see that mut-L can no longer form this closed structure, which is likely what is impacting the disease state. So I'll go through this pretty quickly. So I want to leave a few minutes for questions in case anybody has questions. My graduate student is working on understanding the function of NSP15, which is a non-structural protein that is involved in proliferating the SARS-CoV-2 virus. So a lot of therapeutic strategies have limitations. A lot of them target the viral entry. We're interested in these non-structural proteins, which are made once the virus gets inside of the host. And these NSPs, or non-structural proteins, are intriguing strategies for therapeutics. And so the NSPs are thought to form this replication transcription complex. Here's an atomistic model of that RTC. In the center of it is NSP15, which is the protein that we're studying. Again, this is just a model, but it could be NSP15 might play a very, very central role in this replication transcription complex that helps the virus to evade the host immune response. If you're familiar with the drug remdesivir, it targets NSP12, which of course is one of the important non-structural proteins that might be in this RTC. So my graduate student, Kenya, has designed this FRET and FCS based assay. So we're now using the two together in order to detect cleavage of RNA by NSP15. So here's a cryo-EM reconstruction of NSP15. And so we've used an unlabeled version of this. It's homohexamer, so it's a little bit hard. It's a little bit hard to label. So we've used a labeled substrate. So in this case, we've got a DNA anchor. The donor is on the DNA. The acceptor is at the end of the RNA strand. So this is a hybrid strand. We anneal them together, and we have a cleavage site in the center. So NSP15 should be able to cleave uridine. So it's shown a strong preference for cleaving uridine, 3' of uridine, and it should not cleave A. And so we wanted to design an assay that showed that, and give us a platform to monitor the interactions and maybe look at inhibitors of NSP15 in solution. And so very quickly, with the substrate A, we see no change in that FRET efficiency distribution after some time. So this is after 2 hours. And then for U, we see a complete shift from really high FRET to 0 FRET. And so that indicates to us now that we have this small piece, now the donor and acceptor have been separated and the small piece is diffusing in solution. Now with FCS, we can monitor that small piece over time. And that's exactly what you see here. So we get these two readouts simultaneously, and we use those two together in order to assess different things about the interaction between NSP15 and the RNA. The challenge is that we have lots of subpopulations present in solution over time. So we've had to use this sort of mixed modeling approach to tease out the different populations with FCS. They all have different diffusion coefficients, as you might imagine. Bigger stuff diffuses more slowly, and smaller stuff diffuses quickly. So we were able to do that by looking at, by using a couple of different models together. And we can actually, when we make active site mutations, we can see differences in cleavage for the different active site mutations. So green is wild type. This is again over a course of 2 hours. And then this tryptophan mutant seems to be a bit slower at cleavage, and this agrees with the biochemistry that we know about. Last thing that the last sort of big idea is that we can also monitor the substrate itself. We can look at the dynamics that are occurring in the substrate and see whether or not any of that is important for the RNA NSP15 interaction. And long story short, we think that it is. Manganese has been shown to stimulate NSP15 activity, but it's not found in the crystal structures, or we don't really know where it is or what it's doing. But the data that we have currently suggests that the manganese or divalent cations are interacting with the substrate and imparting some flexibility. And it does so in a way that doesn't impact the A substrate as much as it does the U. So we think that there's something very different going on with the divalent cation, and specifically the U base that imparts some flexibility that promotes the interaction between NSP15 and the RNA substrate. Last thing, we are putting these substrates on the surface. So we really are interested in what is the interaction between the NSP15 and the RNA. Right now, we're not seeing a lot that happens in the NSP15. We haven't seen a lot of bound fraction. We want to see exactly how, we want to see real binding events. We want to see NSP15 interacting with the RNA. It seems to be very, very quick, so we want to see if we can monitor that from a surface attached experiment. So these are just some images of single molecules or single complexes attached to the surface, those same substrates that we're looking at in solution, and we are able to click on them and look at the individual intensity versus time traces. So we're still working on this part of the experiment. We're trying to work on labeling the protein, which again is a bit challenging. So in conclusion, we combined this PyFret and FCS together to allow us to monitor different aspects of monitor cleavage and track the different subpopulations in solution over time. And we want to ultimately challenge this assay with different biologically relevant targets and potentially inhibitors for NSP15. So with that, I'd like to thank you all for your attention. I'd like to thank my lab and thank the organizers, and I'll take questions.
Hi, great talk. I have a question. So in the disease state colorectal story, so what was so it can't go into a closed state, so it can't do the repair. Is that right?
Yeah, that's what I would think. I mean, so I think, so those 4 conformations have to be, or we believe must be functional to do a particular part of its job. So, you know, the mutl is able to, it has these flexible linker arms, so it's able to adopt these distinct conformations, and they all have to be functional in some way, is what we believe. So with the one-arm state, the picture that we have is that, you know, one domain is interacting with mut-s, and then the other domain is sort of pulling DNA in towards the complex. So that's the one-arm state, but you can imagine that if it can't close at all, then it would be unable to do that, and it would likely be there's some evidence as well that there's it forms, you know, this closed loop, and it can slide along the DNA. So it wouldn't be able to do any of those things if it were just N-terminal domains were unable to associate.
So when people have those mutations, do they often have just like a insanely high number of mutations in colorectal cancer?
That's a good question. I don't know as much about all of the genomic data.
That'd be cool
Yeah,
to see.
Yeah, that's a good question. Yeah.
Thank you.
Thank you for your question.